Hello, everyone, and welcome to the introduction to the Heritable Hazards of Smoking workshop. This is a re-recording of the original because we had some audio problems with the original. Um, this workshop took place on September 19th, 2019 in Washington, D.C. as part of the annual Environmental Mutagenesis and Genomics Society conference. Why are we here? We know that smoking is dangerous. We've known that for a long time. We know that in particular with reference to our organs and somatic cells, for example, cancer, lung cancer, emphysema, strokes, tooth decay, many pathologies are related to smoking. We also have become increasingly aware of the risks to fetuses if a mother is smoking. And here are some of those. But unfortunately, this understanding we have about the toxicity of tobacco has really not reached all the way down to what are probably our most vulnerable cells, which are our germ cells. We tend to communicate risks, we tend to research risks, and we tend to regulate risks around somatic effects, and there is actually very little attention given to potential germ cell effects. So, why this workshop? Well, unfortunately, there's been mounting evidence that tobacco's toxicity does extend to germ cells, and not only with respect to things like sperm morphology, sperm concentration, but also in terms of the heritable impacts, that is, possible phenotypes in the offspring born of the tobacco-exposed germ cells. We've seen mounting evidence in mammals, and we've seen some in human studies as well, particularly in recent years. And there are multiple layers of risk and multiple products of concern. So, for example, we have cigarettes, we have e-cigarettes and vaping, cannabis, and there are multiple toxicants in each product. There are many vulnerable phases along the human life cycle, beginning with a primordial germ cell through gametogenesis into fertilization, post-fertilization embryo, fetus, and then, of course, childhood, puberty, and adulthood. And each phase features different sets of molecular vulnerabilities. And in each phase, there are multiple mechanisms at play: de novo mutation, mosaicism, chromatin damage, epigenetic errors, or even other things like mitochondrial effects. Now, for the adverse outcomes that could result in the progeny, we've seen a variety of effects in both human and animal studies. Those include asthma, allergies, hyperactivity or ADHD, neurodevelopmental problems, cancer, and fertility, or fertility impairment. And it's also important to keep in mind that we really should think of this as having two dimensions in public health. First, a rear view orientation that looks back on the history of smoking, particularly very heavy maternal smoking, which peaked in the mid-1960s, thinking about what effects are we seeing in the public and children today that resulted from germ cell exposures perhaps quite a long time ago. And also we have to think forward, right? What are the potential long-term effects of the smoking and vaping and other activities we see today that are related to tobacco or tobacco products? The morning featured the background about germline exposures, mechanisms of toxicity, which include de novo mutations and epigenetic errors. After the break, we learned about empirical evidence from both experimental models in mammals and epidemiology. After lunch, we had a presentation about e-cigarettes from Ilona Jaspers, which was not germline focused, but instead brought up broad issues that may have relevance to our discussion. And then we had discussants and an open discussion, all with a purpose to create a position paper that will be published. All right, so with that, thank you. And all of these videos are featured on this YouTube channel.